All right, I think we'll get started as people are wandering in with the next session. So things are I think moving right along and very well organized. So on behalf of Katerina Jameson and the whole team here, I really also want to thank the people who helped organize this, in particular Michelle Ghani, who's spent a lot of time and effort getting all these pieces in place. So, if anything that we need, let us know. So we're going to start the next session, which is on cell therapy, which is obviously a key part and interest in regenerative medicine. Unfortunately, as Kat mentioned briefly, Gay Crooks from UCLA called me up last night, and she was literally at her hotel and had to turn around and go back home because of some family issues. So she can't make it, and hopefully we can hear her another time. Our speakers, who I'll introduce more in a minute, though, are Koji Ito and Doug Sipp. So we actually have an all-Japanese flavor for this session. Just in terms of 2 slides of introduction for the cell therapy work, because not everybody keeps track of this. I put up this slide, which is from a review we wrote now 2+ years ago, so it may be out of date, but this is a list of clinical trials that are in progress or have been in progress using human pluripotent stem cell derived cells. So if people ask, when are those cells going to actually be used to treat patients, they are, and they have been for many years. And so this really highlights what's going on in this field. They're being used ES cell-derived oligodendrocytes and related neuronal cells that are being used for spinal cord repair. Several groups making pigmented retinal epithelial cells for various types of macular degeneration. There's actually a group in France that's been doing ES-derived cardiomyocyte progenitor cells. ViaCyte, who's here in town, that's doing ESL-derived endocrine cells for diabetes. And if I missed anybody, I apologize. And this, again, may not be fully up to date, but I really wanted to make sure that people realize, and in no small part to the work of CIRM and a number of investigators in California and elsewhere, is really getting these into trials. There have been, I'm not going to go through the reports about safety and some efficacy, but some of these are actually not only safe, but seem to be giving benefit. So I think we're going to hear from Koji about another blood iPS-derived blood cells for platelets and megakaryocytes, and anything that's pluripotent stem cell-derived blood cells is, of course, the most important cells that we should be making. So to do this, though, we have to make the cells, and I'm just doing a very pointed advertising for our UCSD Advanced Cell Therapy Laboratory that is now I've said this for a couple months, but in the last month of finalization to get our GMP facility ready. It's just 2 rooms, but we're already talking about expansion. We have a number of trials, cell products that are in line to use this facility. If people want to know where it is, it's in, if you know where the UCSD Calm building is, on Campus Point Drive, or if you want more information, you can look at our website. And we are, more than happy to engage with other investigators, both academic and industry, who are interested in GMP cell production, and we can then connect directly to the UCSD Medical Center, the CIRM Alpha Stem Cell Clinic, to facilitate these trials, literally all the way from bench to bedside. So that's where we're going here at UCSD. So the first speaker that we have today is actually somebody with San Diego ties, Koji Ito, who's now professor of medicine and deputy director of CIRA in Kyoto, and he's a professor at University of Kyoto. I've known Koji for quite a while because, again, he's also interested in pluripotent stem cell-derived blood cells. Before he got into that, he was here in San Diego as a postdoc in Sandy Steele's lab when he was at Scripps, and I think has maintained close ties to the San Diego research community. And again, he's going to talk to us today about his work on iPS-derived megakaryocyte and platelet production, which I think is really a great example of using all our knowledge and tools from basic cell biology, stem cell biology, hematopoiesis, and then also combining this with bioengineering systems to actually scale up the number of cells that can be produced that are needed for platelet transfusions.